Okay, so good morning, everyone. And I'd like to start by thanking the organizers, and in particular, Ilya, for his hard work and his dedication to put this conference on, and also for the opportunity to share with you what I'm doing as a vascular biologist about aging. My work, in fact, stems from a very old adage that says that a man is old as his arteries, from the 17th century. This Thomas Sydenham, just by hearing the heart of his patients and measuring their pulse, he understood that biological age is not the same as the chronological age, and that healthy arteries contribute a lot to the usefulness and the longevity, regardless of the chronological age. And this is not surprising for vascular biologists, but I think you don't have to be a vascular biologist to appreciate how this huge organ, by the way, this is a cast of the vasculature of a young adult, this huge vasculature supports literally all the functions, the other organs. And not only because these are tubes that provide all the bloodborne substances, including hormones, to ensure the cohesion with the organ functions, but also because, as we heard yesterday from Uri Alon, these are the highways that are used for the immune system during inflammation or during immune surveillance, and the point of entry is the capillaries to enter into these organs. And also because we and others have shown that the endothelial cells that are lining the interior of the capillaries, they are producing what we call endocrine factors, which are secreted proteins that are important for maintaining stem cells, for example, or are involved in the process of regeneration of parenchymal cells. And what is aging if not losing these two properties, maintaining the regeneration potential? So it seems that the vascular aging appears poised to play a critical role in the overall physiological aging of the body. And maybe if we find a way to maintain these vascular functions, we could delay age-associated diseases. So how the vasculature is aging, I'm sure you all familiar with what happens to your large blood vessels, the arteries with the atherosclerosis, aneurysm, the loss of elasticity. All this is recognized as a factor for age-related disease and can be corrected by medication or lifestyle. Less is known about what happens to the microvasculature, which are these little tiny capillaries that are the ones that are in contact with our cells in the body, in the different organs. And one of the most prominent phenotypes that we see in aged tissues is the decrease of the microvascular density that in mice we can easily visualize by just injecting a fluorescent antibody that recognizes specific proteins in the lumen of the capillaries. And as you can see here in aged mice, you can see a decrease in the microvascular density. And by the way, it is also true for humans, because if you look, for example, at the dermis of the skin of a young adult, 20 years old, you'll see a lot of very nice capillary network, which is reduced in the skin of older persons. And it's true for almost all of our organs in the body. So the idea that we had is to find a way to maintain this capillary density. And one of the proteins that is hypoxic regulated and is very important, that's why Professor Elekeshet, my postdoc mentor, name it the guardian of the vascular homeostasis, because VEGF is involved in all aspects of angiogenesis, of the formation of new blood vessels. It is also indispensable to maintain some organ-specific phenotype of the endothelial cells. And if we block the VEGF signaling in the heart, for example, of a mice, you can see here this is the capillaries visualized by immunostaining. They will decrease if we block VEGF after a while, and the restoration of perfusion will be done only if we release this blockade. So what means that there is no backup to VEGF. So indeed, we checked the VEGF signaling in aged mice, and it's also true for human tissues, and we could see that the phosphorylation of the main receptor of this signaling pathway is reduced in aged tissue, as you can see here. And so we devise a system, a transgenic system, where we can increase or maintain, this is the right word, to maintain a right level of VEGF signaling. I don't bother with the details. And we were able to increase a little bit more VEGF in the liver of these mice, which is secreted then in the circulation, where we can measure it and can impact the vasculature and other cells in other organs. And here I would like just to bring your attention to two things. One, the dosage. The dosage of VEGF is very important. We don't want to shake the system by huge amount of VEGF. We do that twice the amount of physiological levels. And the second is that we treat the mice, the system is on from 8 months and onward. So all what I will describe to you is about prevention. Yesterday we heard someone asking about what can we do with young people so that they will age, where they will have a delay in their aging. And we are now working also on initiating this treatment when the disease are already there to see if VEGF can reverse some of the phenotype. So the system was enough to maintain a young foal-like level of VEGF signaling, and that was enough to maintain the microvascular density. As you can see in the liver, which is okay, because the VEGF is emanating from the hepatocyte, but also in remote organs, like in the muscle, in the adipose tissue, in the bone marrow, and any organs we looked at. So this work has been published 3 years ago, and I will just rapidly summarize what we have found. And it was extraordinary to see that just by elevating a little bit VEGF in these mice was enough to delay most of the age-associated diseases that we could measure in our lab or by way of collaboration, and I will just rapidly go over it. So, for example, VEGF maintains a young-like body composition and fat distribution, and it's important for the abdominal adipose tissue, which is very inflammatory, but also the adipose tissue in the skin, which is reduced and cause wrinkles and lowered elasticity and everything. So we could see that in VEGF-treated mice, we could maintain a good body composition from lean to fat, and the distribution of the fat was maintained as in young mice. We could also show by placing our mice in metabolic cages that the VEGF-treated mice maintain a young-like metabolism, and if you need more details, I can do that later. And looking at the liver, we could see that the accumulation of fat that happens with age in the liver of mice and human was significantly reduced in VEGF-treated mice, as well as the enzyme that you can measure as a measure to liver injury that were really decreased in VEGF-treated mice. We also saw less senescence in the liver that we heard a lot about yesterday by Valerie. We look at the muscle of these mice, and by different assays, we could show also that there is an increase in the muscle capacity, in the muscle histology, and particularly mitochondria were improved in their number and in their quality. And finally, we looked at osteoporosis, and we saw that both in male and females, the bone mass were really maintained in VEGF-treated mice, and maybe because of a combination of good muscles and good bones, we could also see that the kyphosis of these mice is improved. Kyphosis that we all know when we look at our grandparents. And finally, just because of the time, all these phenotypes culminates in a longer lifespan which is, in my eyes, not so interesting, but more evident and more interesting was the compression of the morbidity time. And if you remember, yesterday we discussed that to say that if we just increase the lifespan without compressing the morbidity period, this period of bad health, we don't earn much. But I think that with the VEGF treatment, and it's more apparent in females, by the way, we have succeeded in compressing this morbidity, this period where you get all these age-associated diseases. So just to continue, just a word about VEGF and cancer, because when we say VEGF, immediately in the brain of the people, you associate it with cancer, because it's true. Cancer cells produce a lot of VEGF that they need to recruit new blood vessels in order for the tumor to grow and thrive. So we were quite skeptic, because you need much higher level of VEGF to induce to provide the necessary blood vessels for the tumor. But we followed by MRI mice to detect the spontaneous tumors. And as you can see here, in a 24-month-old mouse, in this strain, we have a lot of tumors. They were delayed, or at least not increased in VEGF, in 24-month-old VEGF gain of function mice. And by meticulously look at all the mice that we use for the lifespan study, we could see that VEGF treatment not only not induce spontaneous tumor, increase the rate of spontaneous tumors, but it decreases it. Okay, as you can see here, both in males and females. So now, for the last 3 years, we have looked at mechanism of why this tumor incidence is lowered in these mice. And Priya, together with Jenny Wolinski, who is in the audience, has looked at the immune system. And as I will just summarize this. She was supposed to come with her poster, but she's not here today because she's not feeling well. And what she saw is that first the thymus, which is so important for T cells, is maintained, and which is involuting physiologically in old mice and in aging human. The functional mass of the thymus is preserved in VEGF-treated mice, even at the age of 27 months old, which is very old mice. And together with Yechezkiel Eliyahu from Alon Monsenego's lab in Ben-Gurion University, we could see that the VEGF treatment maintains a healthy thymic output. She could analyze CD4, CD8, recent thymic emigrants, and naive CD4 cells, and she could see that in most of the mice treated by VEGF, we have an increase in the number of these population in the blood, but also in the periphery, which is a good thing, because generally approach that maintain the thymus are not able to maintain the immunity, a good immunity in the periphery. And here we could see that in the spleen or in the lymph node, we have the same increase in naive T cells and recent thymic emigrants. Of course, she looked at the function of these cells, and they are all improved in VEGF-treated mice. And in particular, we could look at the exhausted T cells that are accumulating with age and that are expressing the PD-1 protein, which impairs T cell function. And as you can see here, we have both in the blood, in the lymph node, in the spleen, a decrease in cytotoxic CD8 T cells and CD4 T cells, particularly in the spleen, that do not express this PD-1, so that we'll be able to attack and eliminate tumor cells that express this PD-L1, and the senescent cell that, if you remember, Valerie showed that they also express this PD-L1 ligand. So that can explain why in VEGF mice we see less senescent cells. So I will stop about this VEGF, that counteracting VEGF is promoting healthy aging in mice, and hopefully with the help of Merck, we are producing an NAV, which is FDA approved, to induce VEGF specifically in the liver of these mice, in a way to translate these results to something that we could do in human in the future. So, but I would like to take, I think I have 2 or 5 minutes, 3 minutes, to introduce a new model that I think is important in the aging research, which are organoids. I don't know if you are familiar with what is an organoid, but it's a new technology that has been developed in the lab of Hans Clevers in Holland. And it allows us to take a small biopsy of a human tissue and to process it and to grow it in a special condition in 3 dimensions that allow them to self-organize and to maintain the stem cells of the tissue, so that it enables us to grow this tissue, and here the stem cells are stained in green, to grow this tissue indefinitely and to break it and to expand it and to freeze it and to thaw it and to use it as a regular 2-dimensional tissue, but 2-dimensional culture, but now you have a 3-dimensional that conserve, that maintain the cell-cell interaction and the cell interaction with the extracellular matrix that we heard a lot yesterday about its importance. So having that in mind, people are increasingly using these organoids for many basic and translational applications that are listed here. And most importantly, the FDA has approved this model as a valid preclinical model for drug testing, for example. But for aging, these models are not yet very much used. And one of the problems, I think, is the access to tissues, to human tissues that necessitate Helsinki and etc. So in the lab, we have a lab in Adassa Hospital where we got all the necessary issues to use these human tissues, and we are testing these organoids to validate them as a good model for preclinical for your anti-aging drugs, or just to, if you want to test a pathway, an intrinsic pathway in a specific tissue. And for example, what is interesting is that these organoids maintain their biological age. If you do an organoid from a young patient or an old patient, they will maintain their phenotype in culture, which is huge. And in collaboration with Dudit Sfati from the Hebrew University, we have measured, for example, the telomere length of that in the cells that are grown as organoids in vitro. And we could see that an old organoid has shorter telomeres and a young organoid has longer telomeres, and when you divide these organoids, the telomere will shorten more rapidly than in a human tissue in the body, but it could really mimic the aging of the tissue. In collaboration with Centarix, a company that you will hear, I think, today or tomorrow, we are also exploring the transcriptome differences between these organoids generated from young and old patients, and we already know that these organoids maintain their epigenetic age, which is really nice. And I think that this model can really be used to test anti-aging drugs or pathways that we think are important for the aging science. And we can think also in the future to use these organoids for regenerative medicine. It's not science fiction. It is already used in Japan to repair the intestine of people having refractory ulcers, for example. Experiments are done in mice by the time to correct the retina of people suffering from retinal disease. And we in the lab are doing endometrial organoids to try to correct the endometrium of diseased women. So I think that, I hope that I convince you that organoids are the next model relevant, most relevant for the human to study human aging. And you are invited to contact us if you want to use the organoid that we produce. So to wrap up this lecture, I would like to thank all the people from the Elikeshet Lab that have helped in this study. And I hope I convince you that we can correct the adage, a man is old as his artery, from the 17th century, to a man and a woman are old as their arteries and capillaries for healthy aging. Our collaborators and our fundings, in particular Merck and the Israeli Ministry of Science and Technology, that allows us to translate our results. Thank you. Any questions?
Microphone, please. Yeah. If I look in general for your presentation in VGF, we basically improve function by correcting for the levels of a protein that circulates. So if we look at the presentation at VGF, the main theme was improving function in relation to correcting a circulating protein. A while ago, it was suspected that growth hormone had those effects. So my question is, what do you think are other similar candidates that could be corrected to improve health span?
As a growth, kind of growth factors?
Yeah, I mean...
So for the vasculature, you have other growth factors that can do the job, but again, VEGF is indispensable, because it is involved in all the aspects of neovascularization and also the maintenance of the organ-specific phenotypic structures that you see in endothelial cells, for example, fenestration in the liver or in the pancreas that allows free diffusion of hormones or lipids or things like that. So this fenestration. And what you say is that it's not only correcting the circulating level of VEGF, but the VEGF also in the liver and in the signaling pathways of VEGF. Yeah. In the organs. We see that we have more phosphorylation of the VEGF receptor, probably in endothelial cells, but it's good that you asked this question because VEGF was considered as a vascular endothelial growth factor. But more and more we can see that he has receptors in many other cells, including fibroblasts, which are so important for secreting the extracellular matrix in every connective tissue. Or in immune cells, we have VEGF receptors also. So it's larger. We have begun with only the microvascular density that VEGF can correct or maintain, but we anticipate that the role of VEGF will be beyond all this microvascular density.
It has systemic effects.
It has systemic effects, yeah. Yes. Absolutely, yeah. And the dosage is important. Too much VEGF is not healthy, but the specific dosage is excellent for healthy aging.
Thank you.